Welcome back, ladies and gentlemen. So I have a short lecture for you today on proteins. But before we get started, I do want to review what we discussed last time about carbohydrates. Keep in mind, you're going to see all these macromolecules repeatedly throughout the whole semester. So it is important that we get a firm grasp on them now. Carbohydrates, recall, are sugars, and they use the monomer monosaccharides, which means one sugar. There were three monosaccharides that we talked about: glucose, fructose, and galactose. And all three of those use carbon, hydrogen, and oxygen, typically in a 1:2 ratio, which gives the molecule a hexagon shape. And this is true for most monosaccharides like glucose and galactose, which is why I want you to think of carbohydrates in this way. However, there are some exceptions. Fructose, for example, has a pentagon shape, and there's some others that we'll see with a pentagon shape as we go through the semester. Now, when it comes to the functions of carbohydrates, there are only two. We discussed how they could be used for structure, and I gave you the example of cellulose being used in cell walls and chitin being used in insect exoskeletons. And while you do not need to know chitin, you do need to know cellulose, starch, and glycogen as your three polymers of carbohydrates or polysaccharides. So if you need to review those functions of those, please go back and do so. The other function for a carbohydrate, and the main one I want you to remember, is that they are for immediate energy. So when you eat something like glucose or fructose or galactose, it's going to go into your body and be used in cellular respiration to give you the energy to do things. Okay? So let's go ahead and get started with proteins. Proteins use the monomer amino acids, and remember that monomers are just repeating units that we can attach over and over again to give you big, long molecules. So these amino acids are going to use 5 of your 6 elements of life. And keep in mind that those 6 elements, we use the word CHNOPS to remember those. Well, amino acids are going to use carbon, hydrogen, oxygen, and nitrogen. Some of them are going to use sulfur, but all of them are going to use these 4. So you can think of CHNOS as your word to help you remember the elements in amino acids. There are 21 different types of amino acids. You can see them all here. And they all have the same structure except for that R group. So let me show you what that looks like. Keep in mind that we have carbons here in the corners. Even though they're not listed, they are there. So all amino acids have what we call a carboxyl group on one end. That is a COOH group. And that is a carboxyl group, and we see that on every amino acid. They all also have this amino group, this NH2 group here. And we see that on all amino acids. That's where they get their name from. So that makes the beginning of all of these look very similar. What's different is what's hanging off the ends, and that's what we call the R group or random group. So of these 21 amino acids, 12 of them you can make in your own body. 9 of them are what we call essential because you must get them from what you are eating. So keep in mind how we can attach monomers together. We talked about that last time. Dehydration synthesis puts the molecules together. It's going to do that by pulling out a water molecule. So let me show you how that's going to work here. Let me make this a little bigger for now. Maybe. Okay. So as you can see, I have used these two, threonine and serine. This is threonine, this is serine. We have our carboxyl group, we have our amino group, and if I want to put these together, then I need to pull out a water. So I'm going to take a hydrogen from the amino group and an OH from the carboxyl group, and that's going to give me H2O, and there's my water molecule. So when I pull these out, it's going to open this carbon to be bonded to this nitrogen. And that's what we're seeing here. So this is your threonine, very similar to this one, only we are missing a hydrogen now. We pulled it out. This is your serine. It's turned in a different direction, but it's very similar to this one, but it's missing this OH group. We still have our double bonded oxygen. We still have our amino group. We still have our R group. But when we pulled this off, the carbon was open and it could bond to this nitrogen. This is what we call a peptide bond. A carbon-nitrogen bond is a peptide bond. And if I wanted to link another amino acid, I could pull a hydrogen off of here and link to that nitrogen. So many of these peptide bonds can be put together to give you a polypeptide. Okay? Poly means many, and peptide again is that carbon-nitrogen bond. So if we are to look at our monomer, again, it's amino acids. Here are our elements, and let's take a look at our functions. For carbohydrates, you only have 2 functions, and proteins have many more functions than that. So the number one function I want you to remember for a protein is that they catalyze reactions, which means that they speed them up, make them go faster. And the types of enzymes that, or the types of proteins that do this are called enzymes. And enzymes lower the temperature that a reaction needs to take place so that a reaction can happen at body temperature versus a much higher temperature that would occur without the enzyme. Proteins can also be used for storage. They can bind to molecules and keep them neutral until they need to be used in the cell. Proteins can be embedded in cell membranes and function to transport materials across that membrane. That's going to give you concentrations inside of those membranes of certain chemicals, and that's going to make it easier for these chemical reactions to take place. And that regulates your body and keeps your cells in what we call homeostasis. Homeostasis simply means that the cell is maintaining an internal balance regardless of what's going on outside of that cell. Proteins can be used as hormones. I'm sure you've heard of hormones before. They are messengers from one cell to another. So you can think of one cell sending another cell a text message saying, hey, I need you to do this. And that text message would be the hormone. Proteins can be used as antibodies, and we've heard a lot about antibodies lately. So this is an antibody. They are used to fight against foreign bodies. It's antibody. Okay, and they fight pathogens or infections like bacteria or viruses, such as the coronavirus. And you can see that each antibody has a specific thing that it can look for. Now, similar to carbohydrates, proteins are also involved in structure, but that's the only function that we see in common with our carbohydrates. They can be used as an energy source as a last resort. So for example, if you have ever watched one of those survival shows like Naked and Afraid, and you see the people at the beginning versus at the end, they've lost a lot of weight and a lot of muscle mass. So what's going on is they have burned through their carbohydrates, their sugars, they have burned through their fats, and now as a last resort before you starve, you start burning through your proteins. But again, that is a last resort that your body would go through. Now, proteins are very species specific. So humans have certain proteins that they need. Other organisms have different proteins that they would use. So how do you get your protein? Well, you know that you have to eat it. So let's say that you eat a chicken sandwich for lunch. What happens to that protein in that chicken sandwich? Can you use it directly as it is in that chicken sandwich? Well, no, because you're not a chicken, right? So when that protein goes to your stomach, it is chopped up into individual amino acids, and your body's going to take those individual amino acids and reorder them and restructure them to make your own proteins that will function for you. And we'll talk about that more here in just a second. But think about what that means. Keep in mind that the way we break macromolecules apart is through hydrolysis. So dehydration synthesis puts the molecules together, but hydrolysis tears those molecules apart by adding a water molecule. So if you need to break down fat to lose weight or protein, for example, you absolutely have to have water in your diet, or you cannot break that macromolecule down, okay? And just before we move on, I will point out this is an example of an enzyme structure. This is Rubisco. It is the most common enzyme in the world, and it is used in plants for photosynthesis. So if you look around, I mean, you see a lot of plants out there, and all of them have bukus of this Rubisco in them, aiding in photosynthesis. So protein structure, we have 4 different levels of protein structures. You have your primary structure, which comes together to give you secondary structures. And these secondary structures come together to give you these tertiary structures. You can see that you have a combination of the two. And these tertiary structures can be combined to give you these quaternary structures, or the 4th level of organization. So all of these proteins, you can see an example of one here. Here are your peptide bonds, your carbon, nitrogen. Here are your R groups hanging off the ends. And the sequence of these amino acids is going to be very important. So when we look at primary structures, you can think about these as being straight chains of amino acids, but I don't mean straight chains like a pencil. I mean more like a broken beaded necklace. So you can see these amino acids have been linked together one after another. And the interaction of these amino acids with one another is going to determine the shape that that protein is going to take. So I want you to think about being in class with all of your classmates. If I were to tell you to line up and I pick the order of how you're going to line up, and you have to hold hands with your next door neighbor, then some of you are going to be very happy about your next door neighbor, and others are going to be like, well, you know, I don't really want this person as my next door neighbor. Well, amino acids are very similar in this way. Some amino acids get along very well and some just don't. So if we had you all in a line holding hands and I said, you can't let go of your neighbor, but you can move around the room, then you're probably going to want to get closer to your friends, to the people that you get along better with. And that's what amino acids are going to do. The amino acids that get along very well are going to pull closer together, and the amino acids that do not get along well are going to push each other away. So this is going to lead to certain shapes. And we have our alpha helix and our beta sheets. And if we put these alpha helices and beta sheets together, then we can get what we call a tertiary structure. You can see some beta sheets and alpha helices together here. And the way that these secondary structures get along is going to determine how this tertiary structure is shaped. And if I take these tertiary structures and I put 2 or more of them together, you can see that this one has 3 of them together. Then I get a quaternary structure, or my 4th level of organization. So these tertiary structures are called subunits. And like I said, we can put these subunits together to get what we would call an enzyme, okay? And enzymes are really important for your body. Some of them are going to be soluble in water, some of them are not going to be. And again, that comes back to the way that these amino acids interact with one another, and the way that these tertiary structures are going to interact with one another. So foods that are high in proteins, there are some foods that have all of the amino acids that you need, and those are called complete proteins. Those include things that are mostly from animals or animal products. Animals have to get nutrients from all over the place, and so it's really important that they do have all of the amino acids that they're going to need. Now, there are a couple of exceptions here. We have 2 plants that do have all of the amino acids, soybeans and quinoa, but most plants are incomplete, meaning that they are missing 1 or more amino acids. Now, beans and peas, this make up a family called the legume family. And then grains, you would have your corn, rice, and wheat, among others. So why do I bring this up? Well, if we think back 10,000 years, society has all been based around agriculture. And it's really important that the population can have all of the nutrients that it's going to need. So all societies have been built based on grains and legumes. Now, the reason for this, these legumes have certain amino acids and these grains have certain amino acids. They are not the same between the two, but if you put them together, then you have a complete diet. So in America, our big grain is corn, and beans are our big legume. So all societies, no matter which ones they are, do grow both of these things in order to have all of their nutrition. Here's some more examples of some proteins. We have catalase here, and we'll work with catalase a little later in the semester. Cata means to break down, and ase means enzyme. So catalase is responsible for breaking down hydrogen peroxide in your body. And if you've ever had a cut and poured hydrogen peroxide on that cut, then you will notice that that cut is going to bubble. And the reason for that is this catalase is breaking down that peroxide so that it does not poison your cells, but it will poison the bacteria that do not have the catalase. Now, the peroxide is naturally produced by your body, so you have the catalase in there regardless, so that you can get rid of this natural production of hydrogen peroxide so that it does not poison you. We also have amylase. Amyl means starch, and again, ase means enzyme, so this is the enzyme used to digest starch. And if you remember thinking back to the carbohydrate discussion at the very beginning, then you would have either maybe eaten a cracker or toast, and if you've ever done that and let that sit in your mouth, it breaks down pretty quickly because of this amylase that is in your saliva. It's what's going through and dissolving that starch so that you can use it. Now, most macromolecules break down in your stomach. Carbohydrates are the one macromolecule that start breaking down in your mouth. We also have antibodies. We looked at those already, but anti for against and bodies, foreign bodies, we're fighting against foreign bodies or pathogens, and path means disease. So these would be bacteria and viruses. If we want to test for proteins, which we have already done, you've seen how that works, then we would use biuret solution, and we look for this blue color as a negative indicator, and we look for this purple color as a positive indication of proteins being present in a substance. So when we looked at carbohydrates, I asked you to think about those results and try to write the problem for that experiment. Now what I would like for you to do is look at these results and try to write a hypothesis for these results. And bear in mind that a hypothesis is always written as an if-then statement. If I do this, then this is what I expect to happen. So I'm going to be quiet for just a minute and let you write your hypothesis. And of course, you can pause the video if you need more time, and I'm sure that you do, so feel free to pause. But if I were going to write a hypothesis for these results, I might say, if I test these foods for proteins, then I expect to see 2 and 3 be positive for proteins. Now, if we look at this, a hypothesis does not have to be correct, and mine that I just gave you wasn't. While 2 is positive, 3 is definitely negative for proteins. So keep in mind that it's the way that you write a hypothesis that makes it okay. It's not whether or not you are correct in your hypothesis in the end. So quick little summary slide for proteins. You have your elements and your structure, and I do want you to know the elements. I'm not so concerned that you know the primary, secondary, tertiary, and quaternary structures, but I think it's kind of cool to know, so I did throw it in here. You do need to know the monomer, and there are 21 different amino acids, 9 essential and 11, technically 12, synthesized by the body. Their functions, keep in mind there are many, many functions, but I do not want you to think about proteins as an energy source. That is a last resort energy source. And examples, we talked about rubisco and amylase and catalase, but another one would be carbonic anhydrase. You have ACE for enzyme. This is the enzyme that removes carbon dioxide from your blood and puts it into your lungs so that you can get rid of that carbon dioxide when you exhale and you don't poison yourself. Here's my resources for this PowerPoint. So I hope that you have filled out your note sheet. Please turn that in for your grade and you can use that on your quiz. I hope you learned something important today, and I will see you next time.